bioinformatics engineer, she is contributing towards improving coverage and accuracy, and she remains the escalation point for production issues. We, Hansarians, are eagerly waiting to listen from you. The virtual platform is all yours, Ms. Trisha.
Thank you. I would quickly like to share my screen. Are you able to see it? Is everybody able to see it?
Yes, it is visible.
It is visible.
Okay, sounds good. Thank you. So, hi everyone. I am Trisha Gulati, a 2014 Hansard Model School alumni, and I welcome you all to the Global Connect Bioinformatics session with me. I hope the slides work okay. Okay. Yeah. So a little bit about my background. In 2014, after graduating from school, I was wondering what I wanted to do going forward in life, like every other person. I had given my JEE exam and was waiting for the following counseling sessions. And then in one of those counseling sessions, I was given the opportunity to pursue a Bachelor of Technology degree in Biotechnology from Delhi Technological University. Up until then, I had very little knowledge about biotechnology, and in fact, I was quite scared to pursue a degree in biotechnology because I did not take biology in school. I always thought my foundation wasn't strong enough like the other people, but at the same time, I had this feeling in me that I wanted to do something to improve people's health and lives. And so I took the leap of faith and grabbed that opportunity, and I'm so glad I did that. During the first semester, I was quite intimidated by the coursework because it involved extensive biology, which is something I wasn't familiar with. But then slowly and steadily, I got the hang of it. In the following semesters, the degree became more about learning how we utilize biological systems as technology, and we started applying this knowledge in labs, both dry and wet. At DTU, the coursework was structured in such a way that I had a programming course or lab every semester, and that is something I really like. During the last year, I was taken over by two of my courses. One was computational biology, and the other one was immunology. And in one of the lectures, I was quite disheartened by the stats showing that so many people die in this world because of the adverse side effects of common generic drugs. And that is something nobody even knows about. The regular paracetamol and all the generic drugs that we take, we think they are for our best. And for most of the people, they do work and they are the best. But for some people, they do not work well and they cause many adverse side effects, and people die because of this. And that's something that makes my heart dwell. Like, I feel really sad about that. I've always felt really sad about that. And that's why I wanted to do something about it. I wanted to contribute towards changing that situation. And then I stumbled upon next gen sequencing and precision medicine under the domain of bioinformatics. And that was it. I had found my calling and I decided that this is what I want to do. Bioinformatics is something that would be the best thing for me because, as I said, I liked programming a lot. And I also learned about biology during all those 4 years. So I was like that this is the best combination for me. And this is what I should go for. So the next thing I know, I was applying for master's program in bioinformatics in the US. And then one fine day, I got an admit from one of the world's best universities in bioinformatics, Johns Hopkins University. I mean, who could say no to that? So I was really happy to have gotten that opportunity. And I was like ready to go there, to come here, in fact, and study bioinformatics and learn more about it. During my time at JHU, I learned so much about NGS and precision medicine, and my passion for bioinformatics grew tenfold. I worked with so many great scientists from all over the world on so many NGS related projects. I got to work in the biggest, like Johns Hopkins biggest NGS lab with a great like scientist, and the project was amazing as well. So I was really happy, and I'm really glad I got that opportunity to study at JHU. And I realized that that is something that truly makes me happy. Working towards NGS, contributing to people's health and lives, that is something that has always made me happy, and it is still making me happy. So I'm really glad I'm doing this. So moving on to what is actually bioinformatics. I mean, I'm assuming that only a few of you would actually know what bioinformatics is, or maybe not know what bioinformatics is. But before moving on to my work experience and what I currently do, I would want to give you an insight into bioinformatics, NGS, and precision medicine because that would help you understand what I actually do. Okay, so talking about bioinformatics, what bioinformatics actually is, as the image says, it is actually a marriage between biology and computers. What we do is apply computational techniques for solving biological problems, and for that we can take an example. So in a human body, there are 20,000 to 25,000 genes, and suppose I have a handwritten document with around 1 lakh pages containing information about these genes, their function, their size, etc. And I ask you to find a particular cancer gene, such as TP53, in that document. If you do this with your naked eye, it would take you days, if not months, to find that gene. But suppose you have a Word doc of similar number of pages with the same information, and I ask you to find that gene, you could just do a Control F in a Windows system or a Command F on a MacBook and find it in minutes. Or, in fact, if you have the computational resources, such as a hardware like a hard disk or something, RAM, you could store all this information in a sophisticated table, aka database, and find that gene in nanoseconds. That is the power of bioinformatics. And biology itself has a lot of data. Like there is a lot of data in biology. So I don't know if people here know about permutations and combinations, but in computer science, we have binary digits, 1 or 0. But in biology, we have four bases, four nucleotides, which is A, T, G, C. So if you understand, or if you basically extrapolate the number of permutation and combinations which you could form with four bases instead of just two digits, which is 1 and 0, it could lead to a vast amount of data. And to deal with such a vast amount of data, you need computational techniques, and that's where computer science kicks in and helps biological problems. And that is what basically bioinformatics is. And so that's something I feel is really powerful, dealing with such a vast amount of data using computational techniques. Okay. So next is something I want to talk about is next generation sequencing. And for that, I would like you to watch this video. I hope it works. Let me know if you aren't able to see it. Just a second. I don't think anybody is able to see it yet, right?
No, it's not running at the moment.
Okay.
Yeah.
Yeah. I'll just share my screen quickly. Is it visible now? Is it?
Yeah, yes, it is
It's
visible.
visible. Absolutely.
Okay. Yeah. And are you able to hear it? You are, right?
No, I don't think the sound is there.
Okay. Oh, you're not able to hear it?
No, no, no.
Okay.
से ले रही है?
Does that help?
Not at the moment.
She's gone back to शायद चली गई वापस.
Your microphone is muted, Trisha.
Trying to run the video. Just wait.
Is it better now?
Yeah, but we are yet to hear the sound, Trisha.
Oh, you're still not able to hear the sound?
No,
Oh.
I don't think so.
Okay. I
Bhumika,
have
can we-
unmuted and the volume is, yeah.
Before sharing the video with audio is to be chosen. Please try that. Maybe it will help.
Sorry?
I'm saying, Trisha, that before sharing the video, it has to be, sharing the screen. Share with audio. I hope
Oh,
that will help.
Teams has something like that.
No, we'll help you out. Not a problem. We have the technical
Yeah.
assistance over here. Not a problem. Bhumika,
Yeah, actually,
Please,
once
please.
you click on that sharing icon, the arrow icon,
Yeah.
you'll find
Yeah.
a slider over there. Include computer sounds. So click on that, slide that include computer sound, and then select YouTube from there. Then select video from there.
Yeah, I'm trying to find that option. It doesn't say anything. You said click on the arrow icon, right?
Yeah,
That's
click
where
that.
I share the screen from. Yeah.
Right. And then you'll find include computer sound over there.
I don't see it.
Okay.
Just go slow, Bhumika. Step by
Yeah.
step. Okay? Step by step.
Do I
Is there
have
any
to?
option? Is there any
I
option?
haven't signed in with my account. I've just signed in as a guest. Maybe that's why I'm not seeing that option. Maybe that's the case.
Okay. Any option to include audio? Like, is there anything regarding audio?
No,
Good
there
computers?
isn't anything. No.
Mhm.
Okay.
Trisha, Kavita will help you out. Kavita June. Kavita, will you spell out your assistance? Kavita?
Yes, ma'am.
Yeah,
Am
please
I
do
audible?
spell out.
I can try it once more.
Okay, okay. Or else Kavita has an option.
Yeah, otherwise, Trisha, you can share the link in chat box and Bhumika can share the screen and run it.
Are you able to hear it now?
No.
No.
Not a thing.
Okay, you can share the link in chat box. I think that can work out.
Okay.
Take
Yeah. So.
Okay, give me a minute here.
Children, in the meanwhile, may I request you to call up your friends? This is a wonderful session. Don't miss out these opportunities. You never know where life takes a turn. So all those children who are here, please call up your friends and ask them to join.
Is it any better now?
Sequence an entire human genome.
Yes, now it's clear.
Okay, so I was wearing my earphones. Maybe that's why you were not able to hear
Hello
me.
and
Okay.
welcome to today's
At least
talk
now you're
about
able to
next
hear it.
generation sequencing, which is also known as massively parallel or just deep sequencing. Next generation sequencing has completely revolutionized genome analysis. With next gen sequencing, it is possible to sequence an entire human genome in just one day. We start with the sample preparation. Once we have isolated our DNA, we need to generate smaller fragments. Now all of these DNA fragments will get ligated to certain oligonucleotides. There are two types here. These include sequencing binding sites and also an additional complementary sequence, which can then hybridize to the flow cell. First, our DNA double-stranded fragment is denaturated. So in theory, the whole genome should be in fragments, and all of the fragments have attached two different types of binding sites on each end. All the DNA fragments are now applied on a chip, the so-called flow cell. And as you can see here, there are two types of complementary sequences, the blue ones and the red ones in our example. The DNA fragments will at this ends hybridize with the flow cell as applied to the plate. To make this easier, we just look at one fragment. The first step here on the flow cell is the DNA amplification. By polymerase chain reaction, the complementary sequence to our single-stranded DNA fragment is synthesized. This process, which we have seen here, is done simultaneously for all DNA fragments on the whole flow cell. Now the DNA is denatured, and the single strand, which is not attached to the flow cell's oligonucleotides, gets washed away. The next step is called bridge building. You may have noticed that we have two different types of oligonucleotides here, and the second type of oligo will now also hybridize to one of the sticky oligonucleotides on the plate. This process is called bridge building. The DNA is now amplified again, so a polymerase will synthesize the complementary sequence again. This whole process is called bridge amplification. The DNA is denatured, but now you can see that both fragments stick to the plate. The whole procedure will just repeat now. The DNA fragment will build a bridge again, and again we also have the bridge amplification. That whole process will be repeated for several rounds. And this here is the result. So out of one DNA fragment, we generated multiple copies by PCR. We have reverse and we have forward strands from our copy here. One of these strands here is cleaved now, and sequencing can begin. For sequencing, we just look at one copy now. As mentioned in the beginning, a primer can bind to the oligonucleotides. So this is the sequencing binding site here. Sequencing is now done with specific nucleotides. These nucleotides in this case are fluorescently labeled. Only one of them will fit. In this case, the T. The fluorescently labeled T will then be excited by a laser, and the fluorescent signal is obtained. This is done with a color code. So we know that in this case, blue is T. In the next case, the A will join. Again, the fluorescently labeled A is excited, and we will receive a signal again, this time an A. This whole procedure is done for all of the nucleotides, one by one, and this is how we get the sequence. For simplification purposes, we just looked at one of these copies. Now, notice that due to PCR, we have several copies. So many light signals are obtained simultaneously. The last step now is the data analysis. So here informatic tools are required. The whole process of next-gen sequencing generated millions and billions of reads. These reads are now overlaid and compared to a reference genome. And by this overlay, we can now generate the whole genome sequence. This was it. I thank you very much for watching. I put two interesting videos in the
Able to hear me now?
Yes, you're audible, Trisha.
Okay. Yeah, just a second. I need to change. Okay. Yeah, I think this would be better. Okay. Are you able to see my screen again?
Yes, visible.
Okay.
It's visible.
Okay. So I hope that video must have explained something about NGS. I know these are very like big terms that since you are in school, like you would not know about them. But I hope it explains something. But like I wanted to give an overview about NGS in layman terms. So what we basically do is I have a patient sample. It could be anything, blood, anything that gives me DNA. So what I do with that DNA is I fragment it. I have small pieces of that DNA. And then what I do is I have multiple copies of those small pieces. And then we basically, so you have a puzzle, right? You assemble the pieces. That's what we do with the small fragmented pieces. We assemble them to form a big long fragment, and then we compare it with the reference. So we have a standard that this is what the human genome should look like. We have a reference or a standard, and we compare the fragmented pieces which we have joined together, assembled together against that reference standard genome, and try to identify where is that patient's genome different from the reference, and if it is different, whether it could cause any disease or not. We try to understand if that difference is pathogenic or not. And that is something I basically do, and I would explain that later. But yeah, that's what the motive of NGS is. We want to basically treat a patient according to that patient's genome and not just general stuff that we do, like that we have been doing traditionally. So moving on to the next slide. I hope it's visible. Okay. So next, I wanted to talk about precision medicine. So precision medicine, according to the Precision Medicine Initiative, is an emerging approach for disease treatment and prevention that takes into account individual variability in genes, environment, and lifestyle for each person. So, on that note, I want to mention that all of us are 99.9% genetically same, all of us. It's just because of that 0.1% of difference in our genes or DNA. That is what makes us different. That is what gives us different phenotypes. And by phenotypes, I mean different features. Somebody has a different complexion, somebody has different color of eyes. That 0.1% makes such a huge difference in our phenotypes. So why are we still dwelling on going for the traditional medicine? Like, why are we only thinking about the 99.9%? We need to start thinking about that 0.1% that makes a huge impact on how medicines affect us. So that is what precision medicine is about. We need to be careful, or we need to be mindful of the differences we have as people. And that is what this image shows here. Like in traditional medicine, what we would do for cancer is we would go for chemotherapy, radiation, and that is something that still happens. I mean, traditional medicine hasn't been eradicated. We are working towards precision medicine, but that's something that generally happens. We give the person chemotherapy, radiation, and what that does is it also basically damages or destroys the normal cells as well. And that is not something we wish for. That is not something we want. We don't want to kill the normal cells because they are normal, right? So, what precision medicine does is with precision medicine, I am trying to understand which gene in your body is causing cancer. And if I identify that gene by sequencing it, I can fix that gene. What so suppose that gene sequence should have been ATGC, but in a patient sample it is ATCG. What I could do is with the newer bioinformatics technologies or the genomics technologies, which we have something called gene therapy, we could basically replace that gene with the correct gene, with the correct sequence, which is ATGC, and the problem would be resolved. And that is a more effective way of treating the disease rather than traditionally just giving the person a paracetamol or a painkiller or a comiflam. Like, that is not something we should be doing. So that's what precision medicine is all about, and that is something that really interests me, because I feel like a person should be treated according to their own genome and not like generally. like we do these days. But I'm glad that physicians these days are recommending gene therapy, like NGS sequencing. They are planning to go for NGS because there are so many cases where a physician or a doctor isn't able to understand what's going on with the person. Like you would see these patients for which physicians have ruled out all the common diseases. Like they have ruled out that this person could have this thing. They don't know what actually is going on. And that's when they recommend NGS, sequencing the genome and trying to understand what's going on in the body itself, in the genes itself. So that is what NGS and precision medicine is all about. Another thing that I would want to mention here is that what you saw in the previous slide, NGS, that process, that whole process of fragmentation and assembling and then comparing it against the reference genome, that's just a process. A pipeline is where you decide which tools are you going to use to follow that process. So suppose I want to make a PDF or a Word doc for that matter, I could use Microsoft Word or I could use Google Docs, right? So there are two different tools for doing the same thing. And that is what an NGS pipeline entails. So if I have a pipeline by pipeline, I mean steps. So suppose I am making cake, okay? And in one of the recipes, I'm using sugar, and in another recipe, I'm using jaggery. Okay, that should give me the same output, right? I should still have my cake in the end, but the steps would differ. So what I basically do as of now is that we have these different pipelines, we have these different recipes, but what we want is that they should give us the same result. And that is where my role comes in as a bioinformatics engineer. What I do is, like, I have to understand where are the steps different, and if the results are changing, if the results are not the same, why are they not the same, and what could I possibly do to make them same. And that is what it's called, testing strategy, validation testing. That's what I do. I need to make sure that everything runs well. That is my part. That is the responsibility I have as a bioinformatics engineer. And I'm working at a national lab. So it's a national non-profit and academic reference lab at the forefront of diagnostic medicine. All that I do, these bioinformatics pipelines that I work on, that I test, they all are related to NGS and precision medicine, and basically improving people's health and lives. And another thing that I do is I conduct functional and regression testing for our in-house bioinformatics pipelines. And what that actually means is that, as I said, like, the both the pipelines or both the recipes should give me the same result. What that means is they both have should perform the same function. So that's what I do. I test these pipelines that they should give me the same function, they should perform the same function. And for that, we have several tools. One of the main important things for that is programming. So I need to automate this process. I can't do this with my naked eye. I have to automate this process. And for that, I write Python code to test this and see that everything's working well. That is what I am currently doing. Another thing that I do, as I was introduced with, that I'm the escalation point for production issues. So what production means is that actual clinical patient data. Since it's a national lab, we get clinical patient data, and somebody has to deal with that clinical patient data, right? And we have to do that very quickly because a person's life is at stake. So if one of the pipelines that we have for sequencing fails, or there's something wrong with that pipeline, I'm the person who has to fix that pipeline immediately by finding the bug in the code and trying to fix it as soon as possible. So that's what I do. Previously, what I have done, like, I've worked in a startup. It was the first biotechnology startup in Las Vegas, Nevada. It's called Heligenics. And I was the bioinformatics lead I was the only person who was doing bioinformatics, so you could call me the lead. I was responsible for the NGS pipeline of the company's proprietary assay. So the assay we had, I was responsible for the NGS pipeline. I was building everything from scratch. So that's what I worked on. And another thing that I did there was parallelizing the NGS pipeline. I know these might seem very weird terms, but what that means is, suppose I have 10 steps in a process, and what you would do is that follow step 1, step 2, step 3, and so on and so forth. But if I want it to be done quickly, if I want that process to be finished quickly, what I could do is similar, like from my previous example, if I want to make cake, I could mix cocoa powder, sugar, and everything together once and for all and complete all the steps quickly. And that's what we need to do while we parallelize these pipelines. Since these pipelines are so data intensive, they have so much data to deal with. It could take days, but that's not what we aim for. We want them to be completed in hours. So I used to parallelize these NGS pipelines, containerize them, and make them run quickly, like how to run all the steps together at the same time so that it would be finished quickly. So that's what I have been doing and establishing and employing skills in NGS analysis, pipeline development. So I hope this gives you an insight into bioinformatics. So, well, this brings us to the end of the presentation. I'm very grateful to my alma mater for giving me this opportunity and letting me speak about my journey and passion for bioinformatics. I hope this was insightful for all the students, and maybe someday some of you would become great bioinformaticians. Thank you again. I'm open to any questions that you have, so go ahead.
Thank you so much, Trisha, for such a wonderful and informative session on amalgamation of bio and technology.
Thank
And
you.
without much delay, I would like to invite students for Q&A session. Please raise your virtual hand so that we can invite you for the questions. Yes, Vamsi, please go ahead with your question, beta.
Hi, ma'am. Good morning to you too, and good morning to good night to Trisha, ma'am. Oh,
No worries. Yeah,
yeah.
go ahead.
I was wondering. I came across this whole bioinformatics field in while I was researching for career, next career. But then I went to known when I came to know about bioinformatics, I was scared because right now in 12th, I have like biophysics and chemistry. I don't have computer science, neither do I have math. So like you said, you didn't have the base for bioinformatics. Similarly, I can say that I don't have the base to do the informatics part of the whole project. So yeah, I was kind of worried and confused about that whole part. So clear it up that way.
Yeah, before going to Delhi Technological University, I had no clue about computer science as well. I had no clue about coding. That's something I learned during my during my bachelor's. As I said, like DPU's coursework is structured in a way that we had a programming course or lab every semester. And also, like with time, I've realized that coding is something nobody can teach you. That's something you have to practice, you have to learn. This is something you will have to practice for long hours in order to learn. Nobody can teach you programming, nobody can teach you coding. So if you have the will to learn, you would definitely learn it and be great at it. So that's what you need, the will to learn, which I had. I did not know biology, I did not know computer science. I still took that leap of faith and I went for biotechnology. And that's where I learned everything. And then I went for bioinformatics. So I think if you are willing to learn, it would be a great opportunity for you.
Actually, I have a follow-up question as well. You said you didn't have computer science in 11th or 12th, so
Yeah, I
uh,
didn't.
oh, great. Thank you so much. That helped a lot.
No worries.
Okay, thank you, Vamsi. Next is Kashish Bhatia. Please go ahead with your
Yes,
question.
good morning. Actually, my question is that some colleges make it very clear that you have to study maths or some certain subject for getting a particular course. Like you have to score good in that subject in class 12 boards. So, was there any compulsion in having maths or something like that for applying to this?
Kashish, can we
Do
request you to switch on the video because it looks nice when we ask a question? Let's be face to face. Kashish, Kashish.
I'm on my laptop and there is no webcam and my
Okay.
phone is with my mom.
All right. Fine. Please go ahead. Please go ahead,
Yeah,
Trisha.
Yes, ma'am.
yeah,
So
yeah. Go ahead if you have any
yeah, so
more.
many people have told me that for biotechnology they needed maths and so they didn't apply for it.
N- During my year, when I graduated, there was this thing with JE that 60% weightage was given to the exam itself and 40% weightage was given to your school, like CBSE board percentage. But I don't think that's the case anymore. And all it depends on how you perform in the entrance exam. And if you do well, you can get into anything. So I don't think that matters, the subject.
Okay, thank you.
Mhm.
I'll just add something, Trisha, just for the
Yeah.
information of Kashish and all the others over there. Definitely, you don't have to have full maths, but there is a very strong component of mathematics included in bioinformatics. So, if you have a background of good mathematics and having studied, definitely it will help you. It will
Help,
help.
yeah, definitely.
It
But
will
as
help
I
you.
said, these are the things
Yeah,
you can learn, like
yeah. You
at
can
any
learn
point of
something.
time.
You can pick it up along the way.
Exactly. Yeah. So as I said, like I was scared too that I did not have biology, and people had studied biology for 2 years in their 11th and 12th. But then, like, you pick it up, like, as much, like, the more time you will spend on things, the more you'll learn about them. So it's a will that matters the most.
Yes, I think will and then practice that makes you perfect. Next is
Exactly.
Shifali. Shifali, please go ahead.
Good morning, everyone. My question was that, as you mentioned, ma'am, that first for your undergrad you did biotech from DTU, and for your masters you switched on to bioinformatics. So
Mhm.
both the fields are linked to the base, but they are different to the core. So I wanted to know that was it a change of interest or it just led from one to another?
So biotechnology is the category and bioinformatics is the subcategory. So it all falls under biotechnology and you can then pick up what you want to do. There is a wet lab portion in biotechnology, which is something where you work in the lab, you collect the DNA sample from the patient. Where whenever you go for a blood work or something, those are the people who work in the wet lab. And that's not something I was interested in. I was interested in the dry lab. I liked coding, as I said. Like I practiced through my 4 years of bachelor's. I really liked it. And I mentioned like during the last year of my undergrad, I took up the course computational biology and I was really interested in that. So that's when I decided that I wanted to go for computational biology. And as I said, traditional medicine involves working in the wet lab, but precision medicine, computational medicine involves computers. If you want to go at that scale or at that level of resolving such vast amounts of biological data, you have to go for the computers. You have to go into the field of computer science. And that's when I decided that this is it, that this is what I want to do.
Also, I had a follow-up question that, like, you did bioinformatics in your master's, and as you
Yes.
wanted to integrate, as you said, the coding with the biological thing. But if I pursue bioinformatics in my undergrad only, so would it make a difference that is biotechnology a base for that?
No, it's not. Bioinformatics is specialized like subcategory. Even when you pursue an undergrad in bioinformatics, they would teach you about biotechnology. Even during my master's, in bioinformatics, we had courses related to biotechnology. We were explained everything, like all, like how bioinformatics was even started. It so in order to pursue bioinformatics, you need patient samples, right? Which is something that comes from biotechnology. Biotechnology is in the lab. Most of it is in the lab. So all the patient samples come from the lab. So they explain to you everything from scratch. So you would have the knowledge, but you would have a specialized knowledge in bioinformatics if you pursue an undergrad degree in bioinformatics. So yeah.
Thank you so much. It
Plus,
really helped.
like I want to mention it here that in the wet lab, you have wet lab is a laborious job. Like you have to be in the lab for long hours, right? You have to be in the lab for 8 to 10 hours, and most of it is like standing work. You have to stand and physical labor, and a lot of people are interested in that. I am someone who likes the dry lab part. Like I like coding. I like spending all my I like investing my brain in coding. So whatever works for you, that would be the best.
All of us really wanted to know about these fields. So, as you have explained about them, so it really helps us and it would really help us to fill the forms as well right now. So, thank you so much for that.
No worries, no, not a problem. I'm happy to help.
So nice.
Can I ask a
Now,
question based on the what Kashish asked just now, in continuation with that, Trisha? When you
Mm-hmm.
applied for this university, because children like to apply outside. So did
Mm-hmm.
you take how many options did you have, and where all did you apply? And what
Mm-hmm.
was the criteria for selecting, at level 1, 2, 3, 4? What were the basic parameters on which you focused? जो इन्हें भी मन में रखना चाहिए when they are applying for overseas. Could you just share with
Yeah,
us?
I think the thing that mattered to me the most was where is the course best at. So in the US, there are universities, Ivy League universities, Cornell, and every like Columbia. They are famous for a particular course, right? They can't be famous for all the courses that they teach. For me, the most important thing was that the course that I want to pursue, bioinformatics, should be the best at the university I go for. And Johns Hopkins University is known for medicine, is known for bioinformatics. It is the best course at that university. So that was the criteria I had. And I also looked at the coursework they offered, like all the universities that I applied to. And as I've mentioned, NGS and precision medicine are the two main like categories or things, topics for me that I want to focus on. So, like in my course at Hopkins, they had a specialized NGS course and another follow-up course for NGS data analysis and another follow-up course for algorithms for bioinformatics. So that's what something I focused on, that the coursework should really matter. So that's the criteria I had in my mind. And also, like for some people, budget also matters. Money, taking loans, and all of that. That's a restriction as well. So I understand that. Hopkins is an expensive institute. It is a private university after all. So those are the things you need to consider while choosing a university on what will work best for you in every domain, in every perspective. So
we have one more student, ma'am, Suhani Chanda. Suhani, please go ahead.
Good morning, ma'am. I wanted to ask that the gene that is inserted, will it actually work perfectly or will it have some side effects?
So that's something they're still working on. It's called a gene targeted therapy. So what sometimes I'll I can go into more detail about this. So suppose, as I mentioned, there is this gene that is incorrect. Okay, what you could do is um, there are some regulatory elements that control the production of that gene. So suppose like in a regular human body, the quantity of that gene should be 10 copies. Okay. And because of that, change in nucleotide because of that incorrect gene, the number of copies that are being produced are 5. So the body would not function properly, and that's an issue with the body. What we could do is we could actually, we have technologies. There's one technology called antisense RNA technology, through which I can just replace that incorrect gene with the correct gene. And that's something they're still testing. It's not 100% like valid or something. They are still testing it. It's still in progress. But yes, there are higher chances that it would succeed. Or this is one way. Another thing is that they could change that regulatory element to produce like 10 copies instead of 5 copies, and that would resolve the problem as well. So these are the two like targeted gene therapies that could be there in place. And yes, there are like, nothing is 100% true, right? Everything's in progress right now. And this is a growing field, a developing field. There's so much to learn, there's so much to grow on. So I think soon we would reach that stage where this would be problem free, or maybe almost accurate. So that's what my hope is.
Okay, the last, Tanish, we have one more student with a question. Tanish, please go ahead.
Kavita, we have Kavita Mathur ma'am also. I'm seeing her raising her
I
hand.
am also waiting for her
Actually,
question.
students have
After
joined
Tanish,
through her system. They all are sitting with us. So Tanishk is asking from ma'am's number one.
Good morning, everybody. I wanted to ask that what different colleges of India besides bioinformatics, what other options are available in human genomics?
In India, I think there are, so if you're talking about companies, there are a few companies that I'm aware of. Institutes, I'm not that well aware of, but I think there is one in DU that has a course in bioinformatics. That is a nice one, I think. Other than that, there are institutes specifically. So I did my internship at DRDO. So they have like a bioinformatics department that bioinformatics plus neuroscience that works towards like precision medicine and everything. So I'm not sure if they have academic programs at DRDO, but they do have like internships and jobs at DRDO. So that's what I'm aware of. I always wanted to come to the US because I felt that the scope of bioinformatics at that time was really high in the US, and I'm glad to have that opportunity. But I'm sure there are some colleges, like there are some great colleges for bioinformatics in India as well.
Thank you for sharing, ma'am.
Thank you.
Thank you so much, Trisha, once again. And I would request Principal Ma'am now to share her views on marriage between biology and computers. Very interesting.
I thought Kavita Mathur will come in first. Is she not wanting to ask a question?
No, ma'am. Students only are asking from that
Kavita
account.
Mathur
They are
has
sitting
raised
with
her
ma'am
hand.
only. Kavita Mathu ma'am is sitting with students only.
Yeah, I
The
know.
students
Okay.
are asking their joint
On
project.
behalf. Okay, I got it. I got it. Okay, okay. I know they're all in their lab. They're all here to attend classes. Trisha, indeed, it's been. Ma'am, you're muted. Kavita, ma'am, you're muted. Kavita, ma'am.
Yeah.
Yes, ma'am.
Ma'am, actually, students have so many questions to ask, but Kavita is saying the time is limited, so we can mail her the questions so that they can get
Sure,
their answers.
Yes.
Anna?
I think fair enough.
definitely.
Fair
Yeah.
enough.
Because they actually want to know what different career options are available. They are already studying bioinformatics and this human genomics. This is one very upcoming area. They have studied about biotechnology also in their biology. So they have jotted down so many questions they want to ask, but time is a constraint. So Trisha will definitely mail you the questions, right?
Definitely.
And we'll pass
Yes,
on your answers to the students. Right,
sure,
Vita?
definitely. Yes,
God
yes,
bless you, Vita.
definitely.
So
Thank
nice to
you.
see you.
Thank you so much.
Thank you so much, Trisha. Indeed, heartening to see you come back to the alma mater on a virtual platform. I guess we're doing this for the first time as well to get our alumnus on board. Because vis-a-vis, we would not have been able to get you here. You would be with your mother if you came here to the city. I
Yes,
mean, trying to spend as much time as possible with the family. We understand that. But I guess this mode is really advantageous that way that we get to reach out to you wherever you are. And whatever you said today, though I could not make out 25% of it, it was way above me. I mean,
yeah,
bioinformatics. But I could understand a few things being a science graduate myself. One or two things I can see in the children is a lot of clarifications and a few mindsets that they have regarding this subject and what future holds for them and how important this kind of functional analysis or a structural analysis will have, whether it has a future scope in the career options that they are looking for. I guess they have a lot of questions on this. Should I be taking up anything like this, and what would I be expected to do? And you very nicely said what this kind of a job or a career would entail them, and how much of research and the coding that you do, standing job, the pros and cons of this kind of an option. You very nicely said. I want to add for all the children out there who are, trying to still in a dilemma catch-22 situation, yaha ya waha. One thing I'm sure they want is they want to go out. Great, go
yes,
ahead. But do check out some places here as well in the country. We have a lot of options now than we had a few years ago. My second thing would be do a
definitely.
lot of research before you venture into the area. Everything looks very glamorous from a distance because someone else is doing. This is what they make a mistake in all areas. And she very correctly has told you what will be there in this whole job, in this career, what you will be expected to do. There's a lot of research involved. You'll be submitting a lot of research analysis over there. How important it is what you do to us. If you are doing some kind of a biological literature or whatever you're doing over there, you are working on the data. How important is it? What role will it play in the gene and other protein whatever issues that you have taken up in the analysis of this. ये सब आपको थोड़ा सा आपको ना रिसर्च करना चाहिए before jumping into an area. She has done a good job, but then she faced, I'm sure she must have taken a lot of risks as well. Nothing comes without a, see, she very clearly told you she had no computers background on that. But even then she managed to, work it out to suit her what she wanted to do. Aapko bhi, I guess you'll have to find a way out and see how she has done, and you can do the same as well. So thank you, Trisha, for joining us. I would like to thank Anju Gulati ma'am personally for this, for bringing you on board immediately. And thank you for joining us on this session. और आपके लिए बहुत सारे सवाल और आने वाले हैं. Please do help these children
बिल्कुल,
and mentor them. And they're all alumnus. They're going to be alumnus in a few months from here, and we hope they will do well as well. Thank you for joining
thank
us,
you.
Trisha.
Thank you so much. Thank you. I'm so grateful to you for giving me this opportunity. Nice to talk to you all and let you know about my passion for bioinformatics. So thank you.
Thank you, Madam Principal, for your words of wisdom and for arranging an informative counseling session on such an unexplored interdisciplinary field. I would further like to thank Ms. Trisha from the bottom of my heart for taking time out from your busy schedule and to be the guest speaker at our session. Thank you once again. It was a really nice experiencing interacting with you.
Thank you. Thank you so much.
Thank you and take care. Stay safe.
You too. Thank you. Thank you so much. Bye.
Thank you, ma'am. Thank you, Trisha. Thank you, dear students, teachers. It was a wonderful platform. I'm sure we'll keep going. Thank you once again. Bye from
Thank
India
you.
and a hello to America, right? And keep
Thank you.
connecting. We need more people. Keep
Definitely.
connecting and keep letting us know. Thank you so much.
Thank you.
Thank you,
Thank
Anju
you.
Gulati, ma'am.
Bye.
Thank you. Thank you, Madam Himal, for this wonderful initiative. Thank you, children. Thank you. Now you must take rest now. You must be so tired, right?
Yes.
Thank you. We can hang up
Thank
now.
you.
We can hang up, Bhumika. You can all hang up.
Right, ma'am. Yeah.
Thank you. Thank you. Thank you, everybody.